So look, this series was called From Quantum Bench to Bedside. And the reason we thought it was timely and possibly a little bit earlier than we thought it might have been about a year ago was that following some of the talks last year, people like Margaret and Li Hui, there was evidence of, or we were told about projects which were moving from the pre-normative, commercial phase into the clinic, and possibly with opportunity for therapeutic exploitation down the line. And we heard some of the challenges that were that people were having pushing through their technology, or in fact finding someone to pull it through. And I'll talk a little bit about that later on. So that's why we devised this this series, where we've got actually from soup to nuts in a way, from lab scientists all the way through to those in non-profit and commercial organizations. And it's been good to hear about their work. Most of you will know that on the Roundtable, we've previously asked the speakers to contract their talks down to about 3 slides and remind us what they were telling us about. And because we had 9 speakers and we weren't entirely sure how many of them would be at the Roundtable, what I decided to do, and I hope you don't mind, is I asked Bethany if she would summarize them all for you.
As Jeffrey's already told you, I'll be running through a quick summary of the different presentations we had the series. To all the speakers, please forgive me if I don't communicate your message exactly properly. I tried to stick as closely as possible to your presentations, but it was quite a bit of information to compress. So we started the series with a discussion on electromagnetic fields and cancer. Dr. Jan Corney gave us some insight into the theoretical side of the question. Biological systems require a continuous energy supply in order to maintain their far from equilibrium state. Biological systems are also incredibly complex and need to be controlled, driven, and ordered. So what are some of the forces that might play a role in this? Well, electromagnetism is one mechanism that seems to satisfy the necessary conditions for fast responses across a range of scales. How is this electromagnetic field generated? Well, one of the first to offer a theory was Froelich, who suggested that coherent vibrations were responsible for long-range electrodynamic interactions. There's also evidence that microtubules, which are filamentous cytoskeletal proteins, can generate electromagnetic fields. But what role does this play in cancer? So, as has become apparent so often throughout this series and previous series, one of the key players in the story is the mitochondrion. Jan's research suggests that the electrical properties of mitochondria are very different in healthy and cancerous cells. In cancerous cells, the transport of charged particles such as protons and electrons is disrupted. This results in an increasingly conductive environment around mitochondria and the damping of the electromagnetic field. The hypothesis is that this in turn disrupts longer range ordering forces, causing morphological changes and disrupting communication between healthy tissue and cancer cells. In the context of the theme of this series, how can this theoretical approach lead to downstream medical advances? Well, in diagnostics, current screening techniques don't really use physical properties of cancer cells, which are very different to properties of healthy cells. The damping of the electromagnetic field of a tissue could be used as a diagnostic tool for cancer, but there needs to be more quantitative evidence to improve the use of this technique. Other differences between cancerous and healthy cells, such as field frequency, could also be used as a diagnostic tool to augment current techniques or predict the malignancy of precancerous lesions. On the therapeutic end, fields could be used to inhibit tumor growth or focus heating effects in killing cancer cells. After this focus on the theoretical side of cancer research, Professor Steve Wedge from Cancer Research UK gave us an overview of the current state of cancer research on the developmental side of things. Cancer Research UK is the largest fundraising medical research charity in the world, contributing to the development of new knowledge, as well as bringing 11 new drugs to market. In 2020 and 2021, it spent £421 million on projects ranging from fundamental biological research through to late phase clinical trials. It is also one of the top 3 oncology licenses of the last 5 years. One of the points that Steve emphasized was the collaborative nature of cancer research and development. This involves identifying new areas of research that might translate into new therapeutics, and forming teams with CRUK, but also with biotech or pharma, with the ultimate goal of identifying novel treatments. And this commercial partnership also facilitates late stage investment. In short, forming a research ecosystem that bridges the gap between academia and industry. But despite the challenges that Steve outlined in his presentation, he also emphasized that it is a really exciting time for cancer research, with new insights into tumor cell biology and new technologies to understand and treat cancer. Once again, he stressed the importance of collaboration. Both the approach to cancer and the disease itself is more complex than the idea of a single cure or approach. And CRUK has brought all of its drug discovery and commercialization activity together in a new innovation engine called Cancer Research Horizons. This supports a collaborative approach to cancer research and treatment in order to facilitate the convening of discovery science and its translation into novel therapeutic strategies, with the ultimate aim of delivering breakthrough therapies for cancer patients. In the second session, the focus was on redox modulators, why they are important and how they might be used therapeutically. Professor Alistair Nunn gave us an overview of redox processes, their evolutionary status, and how natural products could be implicated in these processes. I've had to leave out quite a bit of what Alistair touched on, but one of the central messages was around thermodynamics, the dissipation of energy, and the effects of natural products in this context. The transfer of electrons and how this couples to proton transfer is fundamental to the proper function of biological systems in processes such as respiration. Something goes wrong in the transport of electrons through electron transport chains, then this sets in motion a range of adaptive effects or stress responses. Central to this is the idea of hormesis, or a biphasic response to stress. On the one hand, growth, or the other hand, death. And I have simplified that. And once again, the mitochondrion seems to be central to these processes. So how effectively biological systems can dissipate energy through electron transport chains is linked to their capacity for inflammation and aging. The sweet spot would be the balance between too much dissipative stress producing too many reactive oxygen species, which promotes aging, and too little dissipative stress, which acts against the adaptive responses that promote robustness. In summary, as Alistair puts it, the aim is to increase the capacity of the metabolic engine. So how do natural products come into this? Can they be used to modulate dissipative processes? I'll only give a very brief summary of Alistair's more detailed theory. In the development of living systems, a so-called primordial soup gave rise to a number of prebiotic molecules, including chromophores, or light reactive molecules that absorb and dissipate light energy. The structure of these molecules also makes them redox active. This eventually led to the dissipative structures we recognize as life forms today, where the dissipation of energy and the modulation of stress responses are bound together. Thus, natural products which contain these light reactive molecules, such as resveratrol or cannabidiol, might be able to augment and repair these dissipative processes and stress responses when they are used as medicines. So after hearing how evolution shaped and managed energy concerns using natural dissipators such as chromophores, Dr. Wolfgang Brisch gave us a presentation on how synthetic molecules can be adapted towards this end as well. Oxidative stress, often from the overspill of mitochondria, and redox imbalance drive inflammatory pathways, and these upstream signaling mechanisms, such as reactive oxygen species, drive many downstream outcomes, such as mitochondrial dysfunction, cytokines, kinases, and senescent cells. These downstream products are the classical target of conventional medical interventions, such as anti-inflammatories. But these aren't regulatory approaches. Instead of down-regulating the system, you're trying to knock it out altogether, which has unintended and less positive side effects. The strategy is often also immune suppressive, which is potentially problematic. Even more downstream from this, you can see that many diseases are triggered by chronic oxidative stress. So oxidative stress and redox balance is key. For a long time, it was thought that oxidative stress is purely harmful, and the cure is to scavenge all reactive oxygen species for improved health. But reactive oxygen species also play a crucial role in cell signaling. It's thus possibly a bad idea to completely annihilate ROS. The key here, which is still not an accepted approach in pharmacology, is that balance is more important than absolute values of reactive oxygen species. So low doses of natural antioxidants are usually not enough to rebalance, while high-dose synthetic antioxidant approaches eradicate all ROS and throw other processes out of order. The challenge is, how do you target only the damaging effects of ROS while maintaining the useful effects? And this is particularly difficult if you want to treat something like localized inflammation without disrupting system-wide signaling. So Wolfgang and his collaborators have developed a molecule that seems to manage to achieve this ROS rebalancing. The molecule in question is luminol, a chemoluminescent reagent used in a lot of lab work as a reporter molecule. Conventional luminol is not a stable molecule, but Meteopharm has developed a stable version of it. Luminol is inactive at normal physiological ROS levels, but in the presence of a tissue undergoing abnormal oxidative stress, the high ROS levels deprotonate the luminol, activating the molecule and turning it into an electron donor. The activated luminol acts as a ROS scavenger because of its electron donation, and in this way, the luminol is only turned on in inflamed tissues and doesn't disrupt the healthy balance of ROS. There's also an emission of a photon in this activity, which you can see the bottom right, which shows up in the scan of the mouse, which is treated with luminol, only where the active inflammation is located, which is in the knee. So in addition to its effects on different diseases, redox rebalancing using luminol could also allow us to reduce the low-grade inflammation that contributes to aging, leading to extended, if not lifespan, then at least health span. The focus in session 3 was on reinterpreting pharmacological mechanisms through a quantum lens and perhaps using this new understanding to develop entirely new therapeutics. Professor Matthew Fisher began the theoretical end of the debate with a discussion of lithium. Lithium is one of the simplest atoms, has 2 stable isotopes. Lithium-7, which is 3 protons and 4 neutrons, is the naturally abundant isotope, while lithium-6, with only 3 neutrons, is the less common form. More remarkably, lithium is a very effective drug for bipolar disorder, in particular to treatment of mania. It's well established as a medicine, having been FDA approved in the 1970s, but still raises the question of what is the mechanism by which lithium works, and indeed, what is the mechanism by which any psychiatric medications work. Matthew suggests that you need to understand the biochemical underpinning of sentience because psychiatric medications to various degrees modulate our conscious state. So understanding sentience will also allow us to design in a systematic and controlled fashion better psychiatric interventions. Lithium seems a good starting point because it is a very simple chemical and, even more interestingly, a very simple change to its structure, 1 more or less neutron, seems to change its pharmacological effects in experiments on rats. This difference between the isotopes has led to the suggestion that quantum spin might be playing a role, given that one of the few differences between lithium isotopes is that they have different nuclear spin. If the mechanism of lithium's action is quantum, then that suggests sentience in some way employs quantum effects as well. But what physical manifestation does this take? In other words, what could act as a physiological quantum qubit? In the biological context, one of the main arguments against quantum effects in biological systems is the decohering effects of the environment. So to get these quantum effects to last seconds or minutes or hours, which you need for cognition, the quantum objects or qubits need to be isolated. Nuclear spins are best suited to fulfill the necessary isolation. What is a common biological element with a long nuclear spin lifetime? Well, nuclear spins are most effectively isolated when they have spin half, as they're then only sensitive to magnetic fields and not electric fields. So you need an atom that has spin half and that is small enough to tumble in water, which allows it to avoid dipolar relaxation. Beside the proton, phosphorus is the only biochemical atom with spin half. So phosphorus seems to be the only likely candidate for a neural qubit. In biology, phosphorus is mostly found in phosphate, where the phosphorus is bound to and surrounded by oxygen. Oxygen has spin 0, so it doesn't contribute to interacting with the phosphorus spin. But the lifetime of the phosphorus nuclear spin is still quite short because of the interaction with surrounding hydrogen nuclear spins. So you need one more ingredient. You need an ion similar to hydrogen that can outcompete hydrogen in binding to phosphate, but doesn't interact with the spin of the phosphorus nuclei. The best contender is calcium. Calcium phosphate is ubiquitous in the body. It's an important reservoir for calcium and phosphate ions as the main ingredient of bones. Calcium has nuclear spin of 0, so it doesn't interact with phosphorus nuclear spin. And a specific form of calcium phosphate, known as Posner molecules, seem to be ideal for quantum information storage and possible processing. Matthew then went on to talk about where these Posner molecules are likely to be found. Mitochondria are again implicated, where calcium phosphate is an important reservoir of calcium and can affect mitochondrial function. In the second half of this session, Dr. Dave Ecker expanded on how the quantum understanding of physiological processes might benefit medicine. He gave a quick overview of the last new wave of therapeutics developed by him and his colleagues at Ionis. He pioneered the development of oligonucleotides as medicine. So conventional pharmaceuticals generally target proteins. When Ionis first suggested that a pharmaceutical intervention might alter protein synthesis at the source by binding to RNA, the idea met with considerable skepticism. Unmodified oligonucleotides are quickly degraded by the body. Despite this, Ionis, through a concerted investment in medicinal chemistry, developed stable precision genetic medicines to treat a number of diseases. This strategy has proved itself especially potent in the treatment of spinal muscular atrophy, which is a devastating autosomal recessive disease. And this experience of developing a successful medicine from impossible idea to life-saving intervention has led Ionis to question what the next wave of medical innovation might look like. As Dave puts it, understanding life's secrets leads to opportunities to correct life's mistakes, otherwise known as diseases. Each new layer of understanding contributes to our better being able to develop new treatments, from germ therapy leading to antibiotics and vaccines around 100 years ago, to this emerging understanding that there might be another underlying layer to biology that employs quantum effects. How can this be directed towards developing new treatments? In his presentation, Dave outlined 3 approaches that might be employed to use quantum biology to advance medicine. I'll run through them very briefly. First, can diseases with unknown etiologies be explained through an understanding of quantum processes that are foundational to a biological process of life? And furthermore, how do genetic causes interfere with these quantum processes? Second, can we use quantum processes as tools to intervene in diseases that are not necessarily quantum in origin? And this could involve the use of magnetic and electric fields and would potentially address the question of life's preferred frequencies. And finally, could quantum mechanics be used to develop precision genetic medicine? For instance, the use of a quantum trigger could narrow the target of CRISPR technology to a specific disease tissue. Our final session was on light-matter interactions. We began the session with a presentation from Dr. Lise Weber, outlining how Clocks Technologies uses fluorescent light energy, or FLE, a form of photobiomodulation, as a therapeutic. Clocks has completed multiple clinical studies and proof of concept work, demonstrating the effects of FLE on various conditions and diseases. Their lead programs include Floresca for moderate to severe acne and LumiHeal for chronic and post-operative applications. Clocks has attained approvals for FLE in a number of countries. The FLE is generated using a specially formed topical gel or matrix that is LED light activated. This vehicle contains specialized molecules The photoactivation and subsequent emission of these activated molecules is absorbed by photoacceptors within the skin, which in turn modulates various cellular activities, including mitochondrial activity. FLE has been shown to modulate inflammation by reducing pro-inflammatory cytokines. It has also been demonstrated to modulate mitochondrial morphology, activate fission and fusion mechanisms, as well as increasing ATP production. Furthermore, FLE increases important growth factors necessary in wound healing and induces angiogenesis. It also increases and improves the production and deposition of collagen. So Clark's has shown that treatment with fluorescent light energy has long-term persistent clinical effects, benefits, favorable tissue response, demonstrates cell-to-cell interaction and associated healing cascades. They've also shown how mitochondria are implicated in the FLE treatment. But what is still unclear is exactly what is happening when the light first hits the cell. What still seems to be missing, and what Lee's handed over to Professor Greg Scholes to explain, is the physical principle explaining how FLE might function. So Greg's presentation highlighted the collective effects that are integral to something like wound healing or other complex biological processes, where many interdependent networks contribute in an ordered fashion to the overall process. How could one model or frame these collective effects? Greg suggested that synchronization and the models developed to understand synchronization is a useful way to think about biological networks. Synchronization, in which many separate entities act in step, can give rise to emergent behavior. This is a threshold phenomenon where once a certain threshold is passed, something new emerges. There are mathematical ways to model this, which Greg gave some details of. In particular, this model shows what happens if you are given a collection of distributed nodes and are allowed to randomly form connections between these nodes. So adding connections doesn't make all that much difference until at some critical density of connections, a bigger picture or giant graph emerges. At this threshold, things go very quickly from individual behavior to unified collective behavior. This way, collective behavior emerges from how nodes are connected. So how do we apply this to thinking about a biological oscillator model? Well, Greg suggested that we could think of each biological cycle or ordered process as a node. Each node is an oscillator with a given frequency determined by the biological clock for that process. And the nodes are all connected with feedback loops, for example. This gives us enough information to model the synchronization, or alternatively to model how this synchronization is changed or disrupted through tissue damage or disease. And this could also explain how FLE works by restoring this disrupted synchronization, either by modulating the connections between the modes or by modulating the frequencies of the separate nodes. In short, Greg suggested that a way to think about health and disease is not as a measure of the individual entity, such as a cell, for example, but rather to think in terms of a healthy or an out-of-sync system. Our final presentation was from Brent Bourne, who outlined the development of cognitotherapeutics and the science behind it, particularly the emergence of electricity as a new drug. Changing electrical activity can change outcomes and function, and in turn drive protein level changes. In the interests of the theme of the downstream realization of new therapeutics, Cognito has developed a device that uses 40 Hertz flickering light and sound to stimulate electrical activity in the brain in order to treat conditions such as Alzheimer's. Brent presented a variety of really interesting results from preclinical and clinical studies. There isn't enough time to go through all of these, but a sample of the results shows that stimulating electrical activity in the brain, particularly gamma synchrony, activates microglia to clear protein pathologies and promotes blood flow for lymphatic clearance. This in turn leads to reductions in A beta and tau and improved neuron morphology, cognitive function, and circadian rhythms. Gamma synchrony also prevents brain atrophy. Brent concluded by suggesting that gamma synchrony is a novel target space and mechanism for brain treatment. Cognito started with treating Alzheimer's, but is moving towards treating Parkinson's as well. And the aim in the last few years has been to build increasing amounts of data on this novel mechanism.
I'll just have a very, very brief overview here. This is really to stand back a bit and look at what we've actually, so what's being picked out in terms of themes and insights. So what can we glean from the series that we've, this spring series? How the talks provided us with a new way of thinking, and how they led us to an increased interaction between scientists from completely different fields, sparking some original thoughts. And I think most of you know the answer is probably yes to these. Next slide. So just to summarize, in the first session, we were looking at cancer and its treatment. And what seemed to come out from this is we are going to have to embrace some old new ideas. Certainly the idea about electric fields has been around for a very long time, 100 years. But with the modern development of treatments, it's moved very much towards single molecule, single target approaches. It's quite a natural progression because we didn't really understand the electric stuff. But unfortunately, although a lot of progress has been made, and I think Steve has shown how well we've moved forward, we're still a long way from a cure. And in fact, we're still quite a long way from fully understanding what cancer really is. And this suggests that a missing component of this may well be that we need to view cancer slightly from a different perspective. And it could well come through the lens of quantum biology and thermodynamics. And indeed, we may need even to embrace how life started itself to what actually aging is. And certainly one of the things that seemed to come out from me, and certainly this comes from Mike Levine's work as well, is the importance of electric fields in determining self and non-self and shape. And this offers a really quite interesting idea that we can tune, for instance, cancer to cooperate again, and either it will die for the greater good or indeed start cooperating where it should. Of course, this could be, for instance, seen in the immune system and how the immune system is modulated by cancer. So there is a possibility here, by resonating or some form of tuning, we can bring cells back into line. So I mean, again, we're a long way from being able to do this, but it suggests there's another way of looking at it. Next slide. And the second session, we looked at natural products. And as Bethany outlined, one of the key things about natural products, and I mean, these are things that plants have produced for millions of years, is that they certainly evolved to help with dealing with stress. And their quantum structure, again, as Bethany pointed out, seems to help plants deal with stress, in particular phototoxic stress, because their quantum structure enables them to dissipate energy. And in a sense, this plugs into the very basis of what life could be in relation to a thermodynamic principle around dissipation and self-organization. And so if we step back and look at this, we can say, well, actually, through evolution, these compounds have kind of evolved to be involved in multiple different pathways. And indeed, what we're now seeing as we begin to understand these natural products more is that, of course, they display polypharmacy. They interact with multiple systems within the cell, multiple targets. And of course, in Greg's parlance, they're interacting with these nodes. And so you can argue, starting from a very simple basis, they have evolved to adapt to many different systems. And in a sense, we go back to the field idea again. Of course, it suggests that we need to think about how we can use physics to retune these systems. And taking the example of luminol, which is a fascinating example, in a way you can argue that luminol is a compound which can adaptively dissipate energy to restore balance and restore an imbalanced redox system. And I do apologize for the graphs. I'm trying to show this dissipation, redissipation, restructuring thing. So I do apologize. Next slide, please. This leads us on to the really, for me, was of course one of the most amazing things about this idea of Posner molecules and entanglement, because this all goes all the way back to the ideas of consciousness and, is this using quantum principles? But of course, is biology using some kind of informational qubit system? And certainly, if we're thinking about therapy, we may have to start thinking like this and how we might want to modulate this. And is biology actually teaching us something about physics? And of course, on the left here, you see I've copied a picture of what is a qubit computer or quantum computer, which is vastly complicated, but it could well be that life has been doing this for billions of years. And I talk about life doing it for billions of years because has this something to evolve very early in life? But one of the other really important things that came out from this session, especially from David Ecker's talk, is, of course, it's really important that there are companies out there, individuals who are willing to invest in these kind of really original ideas, which is something mainstream science is quite difficult to do at the moment. They don't always invest, but I think if we can find these people, and David is a classic example of this, we really can move things forward very quickly. Next slide, please. And finally, to the light matter. This to me is one of the really building areas that shows that we are actually already getting there with some of this non-chemical modulation of biological systems. And it actually highlights something that Jeffrey has often said, is we don't necessarily need to understand how something works as long as it does, but we've got to prove it works. And this is something Jeffrey will talk about a bit later. And I would also argue, but of course, we can always improve how it works if we understand how it works. And I think this is where the interaction between the quantum physics world and biology is really beginning to show its legs. And really strengthening the interface between these two is absolutely key in moving forward. And I apologize for the little diagram at the bottom. What I'm trying to show there is tired mitochondria and light, resulting in a better ability to run better. So I think I'll now hand over to Jeffrey. Bethany, Jeffrey.
Thank you very much.
Oh yes, the final. Sorry,
Alistair, you were going to do a few more, yeah.
Oh, sorry. I'm sorry. Okay, yeah. Well, the final. How far am I down? How many people have got left, Bethany? Sorry,
3 slides, Alistair.
I've got 3. Okay, I've forgotten an important thing. Yeah, there's a bigger picture here, which we perhaps need to look at, and of course, this is the non-chemical modulation mechanisms. And for me, one of the big factors that come out of all of this is that the need to collaborate and cross-fertilize between different disciplines. And this idea of moving from basic medicinal chemistry to physics and back again is perhaps really far more important than we realize. And just to kind of show the potential collaborations, but they go back a long way, and perhaps in ways that people didn't realize. I'm sure you recognize these two characters here. One's Charles Darwin and the other is Copernicus. Charles Darwin, of course, was famous for his evolutionary theories and natural selection, but of course, even in physics, there is natural selection of molecules. And going vice versa, Copernicus was talking about planetary motion and everything, but of course, that influences evolution and biology. So there's already been these collaborations, perhaps that people didn't realize. Next slide, please. And I apologize for this. I think I modulated and I've slightly destroyed what Bethany did on this slide, but what I've tried to say is what, where, and does it, or is it matter? And of course, this is using matter in 2 different ways, viewing matter in 2 different ways. One is, of course, does it matter in biology that we understand the physics, but also what is matter? And matter, of course, is largely empty space, which from anybody who does quantum physics will tell you, this is what matter actually is. And so really, we've gone from over the last 70 or 80 years from the chemistry of life, and we're moving rapidly to the physics of life, which of course, the physics of life was discussed 100 years ago. So we're kind of going backwards and forwards. And certainly from the talks we've seen, not just in this series, but in the previous series, is how we're beginning to look at photons as quantum entities, but water from a quantum perspective, and water order, long-range interaction. Same with protons, the fascinating role of protons. And of course, electrons is what life's all about. Gurney has always said that life is about electrons looking for a place to rest. And of course, this is entirely what life is about, and he explained redox. And of course, we then move on to Posner and all these ideas about spin. So in a sense, we've gone from chemistry to physics, but we're going back to chemistry and then back to life again. So really, to me, that this is the real biggie here. And this is actually changing the way we view life. And this is about the idea that life is actually almost a thermodynamic certainty when you start thinking about self-organization and dissipation, and the role, of course, how it relies on resonant tuning and vibronics. And if we look at the start of our sort of research journey from 50 or 60 years ago, biology moved towards single target. We've got to get back into single targets. We've got to get the right drug binding the right receptor. Then, of course, over time, people were still thinking about vibration and vibronics and resonance, and from a quantum perspective. And this shifted us to go thinking, well, actually, we need to think about system rebalancing in science, in biology. And here I've got a little picture, which again, thank you, Bethany, for giving the basis of this. Is a game I showed my tired mitochondria on a balance, but of course, tired mitochondria aren't just about any production, but they're actually their inability to modulate redox. And so finding a way of retuning them is actually something absolutely essential. And Jeffrey, Jimmy and I have been talking about mitotuning for about 13, 14 years. But of course, this thinking then brings us back towards multiple targets, because of course, as we mentioned earlier, the role of biology in multiple targets. And suddenly we have to think, well, actually, we can't think about single targets, but we have to think about multiple targets and why biology will be modulating multiple targets. And of course, this brings us neatly back into the vibronic tuning again. So I think that's where I want to finish, but it's important here to think differently about how we approach biology.
So this single slide that I've put up, what I like to do is try and put all the science, both from a physics and a biological point of view, into a context of how can you drive this forwards towards commercial exploitation of therapeutics or diagnostics, therapeutics and diagnostics. And many of you will understand my background, but for those of you who don't, I'm a physician. I've spent 40 years developing medicines. I've developed medicines approved throughout the world through nearly all the regulators, through a whole range of different disciplines, both synthetic medicines, semi-synthetic and plant extracted. And in the process, I've founded 3 public pharmaceutical companies. I took one public to NASDAQ, the next one to London and NASDAQ, and then the last one on NASDAQ. And many of you will know that this time last year, just over a year ago, we sold our last pharmaceutical company that we founded for $7.2 billion. So I have a fair amount of track record in this area. So medicines, and I'm going to talk mainly about medicines, and forgive me, I do understand that there is an awful lot of opportunity for diagnostics procedures as well. But medicines have come from different sources and naissance, both empirical, in other words, observed without a theoretical basis, and from theoretical hypotheses. And over the last 70 or 80 years, most pharmaceutical companies would have considered that their medicines came from theoretical hypotheses, whereas probably a good proportion of them came from empiricism. And when we think about the series that we've looked at so far, it'd be reasonable to say that perhaps luminol and clocks are 2 examples of empiricism. Luminol was first synthesized in 1902, and its use in detecting blood at scenes of crime began to emerge in the 30s with Professor Pfannenstiel in Germany and others. So here's a molecule that's been around for a long time, and I think that Wolfgang and his team have made certain observations and then developed a molecule which could be remain stable and used in medicine. And the same potentially with clocks. And on the other hand, we've heard about, not only this series, but last year as well, 2 emerging technologies that are moving their way into clinic that I think were derived entirely from theoretical basis. And that was Li Hui's work on the 40 hertz cycling of light on molecules initially, and then finally into patients' eyes. And of course, Margaret with her infrared exposure in patients with COVID inflammatory pneumonitis, and to accelerate the healing of lung and inflammatory pneumonitis. And so there's 2 extreme examples: the theoretical the emergence from theory of potential therapies, or observations made over decades that have led to other modalities. And somewhere in the middle there, of course, is the reprocessing. So having an original molecule that's been used in medicine, the lithium, for example, but now rethinking the scientific underpinning of that in terms of it being a Posner molecule. So these are the types of science that lead into candidate therapies. And it's been fair to say that the way you have to take these therapies to get them to common medical use would be you have to go through the regulatory authorities worldwide, and you cannot get around that. In some cases you can, but you cannot get around that. And for the last 70 or 80 years, and certainly the last 40, 50 years, pharmaceuticals has been all to do with single chemicals, supposedly as ligands for single targets. And generally they were soluble. So the pharmaceutical industry was about single soluble chemicals. Whereas we've been looking at, and certainly from a plant origin background, where you get multimodal polypharmacy from single molecules in plants, looking at how these affect systems overall. And interestingly enough, although the scientists in pharmaceutical companies and pharmaceutical companies as a whole like to think about single targets, the regulators are very open to considering systems, and especially in rare disorders. If one presents data on one particular disorder, but the regulators consider it shares the same systems biology, then sometimes labels can be expanded to indications which haven't been proven in a clinical program. That's certainly in very rare disorders, not so much in larger medical disorders. So when you think about a single molecule as the subject or the agent of your therapy, then the regulators are well versed, and so are the pharmaceutical companies and the universities that assist in these developments, in how to develop, assess, evaluate, measure single molecules, how to characterize them both pharmaceutically, pharmacologically, toxicologically, and to generate information on quality, safety, and efficacy. That's a well-trodden path. But as soon as you start to talk about multimodal molecules, some of the preclinical testing that has to be done is different, and the regulator's approach is different, and one has to go back to fundamental principles sometimes. And I'll come back to that a little bit further on. Now we've got this other thing to think about, and I certainly thinking about the comments that Brent made in his presentation, is the science that we're talking about now could fall into one or other of the categories of devices or medicines. And each of those have their own regulatory route, their own burden of evidentiary proof, and their own cost and structures, and their own business models in a way. So I don't think that we would doubt that the 40 hertz light and the infrared is a device, and we probably wouldn't doubt that luminol is a medicine. But Lise shared with us what I thought was a little bit of a bombshell, I thought was marvelous, is that clox is a topical application of a gel to the skin, and that would normally fall into the category of a medicine. But it appears that the regulators requested from clox, does their gel work when the gel is isolated from the skin? And I believe, Lise, correct me if I'm wrong on this, but you applied the gel to a barrier, which was glass, I think, and obtained the same outcome. So is clox now a medicine or a device? And it begs the question is, should you apply the gel to the skin or to the device emitting the light? So these questions will come up quite frequently now, I think, in terms of any application of quantum principles or quantum effects in biology. So in your laboratories, both in university and in commercial environments, and in the non-profits, inventions will arise. Now I'm the named inventor on over 800 patents, which fall within 120 patent families. And there's quite an art to ensuring that what you've invented is something that can be protected. Now, for young biotech starting out, one of the first things they need to know, not only can they protect their invention, but do they have freedom to operate? And you have this dead space in time where you have no idea what somebody else has patented. So sometimes you have to set off down a route for 1 or 2 years, investing significant amounts of money behind your own invention, only to find someone has got an earlier priority date on effectively the same invention, or something that provides prior art and invalidates your patent. So you've got that risk in terms of patenting. The reason I talk about patenting is if you can't protect your invention, it's difficult to fund its advance towards clinical exploitation. The other issue that arises when you have new scientific input into therapeutics is the scientists that are not particularly familiar with the downstream of their upstream invention will quite often describe their invention incorrectly, or they'll share it at a lunchtime journal club meeting and make an unauthorized disclosure, and therefore mean that their invention is the patent is invalidated. Or more commonly, what we see is a series of patents arrive at the same time, where effectively the invention in one cancels out the invention in the other. And so one has to be extremely careful with your patenting strategy to ensure that the invention you make and the claims that you make are borne out in the actual therapeutic that you finally end up with and you want to take to the market. And sometimes that's lost. When I was in one of my earlier companies, we were pursued in 14 different countries by a competitor trying to invalidate our patent. We won in every single country, but what they didn't understand is they were pursuing the wrong patent. It wasn't the one that covered our therapy, which so always you have to make sure your claims will feed through to the therapeutic indications that might come out, or the diagnostic ones as well. Now I come back to testing evaluation. In any clinical indication, there are a range of standard, well understood tests, laboratory models, preclinical models for your target indication. But what you have to bear in mind quite often, very often, is that model is only really valid for a certain class of drug or class of intervention. And that if you move to a novel class of intervention, the test may no longer be valid, and the regulators know that. So you have to go back and validate your models. So when we started to go to the regulators with whole plant extracts, the first in modern times, really, the regulators said to us, well, how are you going to test this? They didn't know. So we had to generate a whole series of tests. Instead of testing for a single chemical on a single target, we were putting forward extracts with 400 chemicals in. And how are we going to test those for their content and for the consistency of production? And it's the same, and I think there will be some real challenges if the intervention is one using or enhancing or harnessing quantum effects in biology. There's going to be a sort of a deadly silence from the regulators who are going to say, what is it that's actually the agent here, and how are you going to test it, and how are you, for example, going to apply a notion of a dose? When I was a medical student, I was told the correct dose is enough, or enough to cover a dime every morning was the other one, I think people said. So we do need to think that when you make a new invention, which is novel for perhaps even a known indication or a very rare indication, you also have to think about how are you going to test it, and how can you justify in front of the regulators that your test is testing what they want to see? And how can you stress test it? The regulators always want to see some results outside the norm. If every single result you put forward in your testing is within the norm, they won't believe the test is sensitive or specific enough. Then we had the question in a couple of our papers in the series is in terms of trials methodology. Now, with a device that is emitting light or radiation from a box or something, one could have a sham exposure. And the same possibly with your gels and other things. But trials methodology is going to be really important. 50%, the regulators tell you 50% of all phase 3 trials fail. And it's not 50% of the drugs failing, it's the trials are failing. And therefore, early in, if one is going to move some of these newer interventions forwards into large-scale trials, much work will need to be done in ensuring that the trial has any chance whatsoever of demonstrating the desired outcome. And I've developed medicines in the past that work perfectly well and ended up spending $200 million on phase 3 trial programs which have failed. And the regulators know that, and interesting enough, all the funders know that. And then finally we come to labeling. At the end of the day, when you put forward your data to a regulator, what you're actually doing is justifying what is in the label. You can only put in the label that which you have demonstrated to the satisfaction of the regulators. If a doctor uses your therapy or intervention outside that label, that's up to the physician. But the pharmaceutical company or the devices company cannot make a claim which is outside the label, and the label is based entirely on the information provided. So when you go through all of these steps, at the end of the day, all of those steps have to lead to a label which is supported by evidence, which hopefully is using the intellectual proprietary rights that you've produced to get there, and is of commercial attractiveness. And that brings me on to my final point, which is to do with funding. The classical or conventional routes of funding to drive programs from the top of this list through to the bottom are very, very much orientated towards the conventional approach of the last 6 or 7 decades. And you see, fantastic funding that, for example, the cancer research puts forwards, and have brought forward some marvelous medicines through that route. Some of you already have funding, and I think David Ecker said, look, they were very successful on one other medicine or in another area of medicine. So could they apply some of the largesse from that to new thinking? That's helpful. The large not-for-profits are raising their money in different directions, but the small startup companies will all have to deal with the conventional funding routes that generally understand the milestones that are determined by these and other bullet points, but generally understand those. And if you start off with something which is born out of a theoretical hypothesis which very few people understand, which seems to be working through systems biology, you're not quite sure it's a device or a medicine. The patent territory is perhaps a little bit unknown. You haven't got a well-established or accepted range of tests or evaluations for it, and with very little understanding of how you're going to carry out your late-stage clinical trials, you can understand why the conventional funders in this route, in this area, may be quite shy of coming forward with their funding.